Hello everyone. Welcome back to Dr. Lou's VCE Biological channel. Today, let's learn together about the 2023 NHT Biology. We will focus on questions related to photosynthesis, cellular respiration, and biotechnological applications. My explanations may need to be revised. If any teachers, parents, or classmates find errors, please feel free to provide your guidance. The function of pigment is capture of light energy. So, inserting accessory pigments could enhance the capture of light energy, especially visible green light. The keywords are light independent. In the light independent, glucose, ADP, and NAD+ are produced. Protons cross the thylakoid membrane, not the inner membrane. Yes, the light-dependent reaction is located on the thylakoid membranes. Oxygen is produced from the water, not carbon dioxide. Light-dependent reactions produce oxygen, ATP, and NAD+. Rubisco is one of the enzymes of the Calvin cycle. Calvin cycle is in the stroma. So, Rubisco is in location D. Glycolysis is located in the cytosol, Krebs cycle matrix of mitochondria, and electron transport on the infer membrane of mitochondria. R is a hydrogen ion. S is not water because no hydrogen is involved here. The electron transport is on the inner mitochondrial membrane. Yes, T is ATP. When the hydrogen ions flow through the channel, ATP is produced. So, D is correct. A non-competitive inhibitor binds to a site on the enzyme away from the active site, causing a change to the active site. This results in the substrate no longer binding to the enzyme, the rate of product formation being lower, or the enzyme cannot catalyze the reaction as efficiently. Human kidney cells are cultured at 5 temperatures. At each temperature, there are 2 incubators, oxygen and no oxygen. Glucose consumption is measured. In the presence of oxygen, glucose consumption at 35 degrees Celsius is 4 millimoles/24 hours. Lactate production at 35 degrees Celsius is 6 millimoles/24 hours. When there is no oxygen, glucose/pyruvate is converted into only lactate. Double the amount of lactate is produced for every glucose consumed through glycolysis and fermentation, or more glucose will need to be consumed as less ATP/energy is produced. When oxygen is present, glucose/pyruvate can be converted into water and carbon dioxide. This leads to a smaller ratio of lactate formed than glucose consumed, or a greater amount of ATP/energy is produced through aerobic cellular respiration to meet its energy requirements. At 45 degrees Celsius, enzymes involved in biochemical processes begin to denature, resulting in a decreased reaction rate compared to when there is optimal glucose consumption and lactate production at 35 degrees Celsius. The photosynthetic output is investigated. The output is the quantity of glucose produced. The plants are exposed to differing environmental conditions, temperature, carbon oxide, and relative humidity. The plant is likely either a C3 or C4, but not CAM. For C3, the quantity of glucose production was higher at 25 degrees Celsius when a higher percentage of carbon dioxide or humidity was present. Higher temperatures increase the affinity of Rubisco for oxygen, leading to an increased rate of photorespiration. For C4, they are adapted to hot environments at temperatures 25 degrees Celsius or above. It cannot be a CAM plant as these are adapted to hot conditions with greater glucose production at 40 degrees Celsius temperatures and above, or low humidity, which was not observed from the scientists' results. CRISPR-Cas9 is used to develop high-yielding rice. The target is the OSHX1 gene. This gene is knocked out in HHZ, CH1, and CH5. The average height and average grain are investigated. The two dependent variables are the height of the plant and the grain yield. The experimenter measures these as a result of changes in the rice variety. HHZ expresses the OSHX1 gene, and CH1 and CH5 don't express OSHX1 because this gene is knocked out. A decreased height and grain yield increased in modified rice varieties. More glucose would be produced, resulting in an increased photosynthetic efficiency. Any two factors are okay. These factors include light absorption or wavelength of light-absorbed carbon dioxide, consumed oxygen production, glucose production, concentration of starch, amount of chlorophyll, water production. The independent variable is the different concentrations of carbon dioxide applied to each plant. The dependent variable is the average grain yield measured. The controlled variables include the same wavelength of light/temperature/humidity, pH of soil/number of seeds for each plant, the volume of water applied to plants. The control group would be a plant exposed to normal atmospheric carbon dioxide concentration. A large sample size for each carbon dioxide concentration is tested. In photosynthesis, cellular respiration, and biotechnological applications, 6 multiple-choice questions are worth 6 marks, and 3 short-answer questions are worth 27 marks. The overall score for this section is 33 marks. With a total possible score of 120 points, the percentage for this section is 27.5% of the total score.
If you need my help, please add me on WeChat or WhatsApp, and send me an email. Thanks for your attention. Please note Dr. Lou's VCE Physics channel, Dr. Lou's VCE Chemistry channel, and Dr. Lou's VCE Biology channel.